So today I would like to introduce our recent paper on report progress physics about computational methods and diffusion theory in triangulation sensing to model neuronal navigation. This is a work, joint collaboration with Ulrich, Dobramysl at University of Oxford and myself. So let me first introduce the question that motivated our approach is how neurons find their path and what happened when they could be deregulated. And so for this, I would like to remind you that neurons in the brain are usually creating connections between various different regions, as beautifully illustrated here with very colorful images. So in practice, and this is an example here for thalamocortical axons, where they have to start somewhere in the thalamus, continue their path, go through some corridors, pass different regions of the brain, and move on to the cortex. So if for some reason a gradient of morphogens, called slit morphogen, is dysregulated, then neurons are misrouted. That is, they are going to go to different directions, which can cause various problems of brain wiring and has a consequence for brain function. Another example is here the two gradients called the neutron and slit gradient. Under normal conditions, these two types of population of neurons are well separated. They are sorted at different locations of the cortex. However, when those two gradients do not exist anymore, then the two types of neurons are completely mixed, which create an incorrect wiring of the brain. So another example of how neurons have to find their path is when you think about the ganglion cell. So in the retina you have 3 layers of neuron, and the last one, the ganglion cell here, the axons are going to project, as we can see here, into a region of the brain called lateral geniculus. And this projection here, and a representation, abstract representation of this projection, is here where you have the retina, where we have the temporal and the nasal direction, and they are projected to the tectum in this anterior posterior, and this structure is actually labeled by this angle gradient. So once you have this patterning as a gradient, the nasal neuron will mostly project to the posterior part, while the temporal one are going to project to the anterior, creating a gradient of location where neurons have to stop. So in other words, neurons should be able not only to sense a gradient, but to interpret the gradient and to stop at the right location. And as we've seen before, they should probably be able to identify where is the source of gradient in order to generate information that will tell them what is their position. So this is another example where you have this morphogen gradient, this sonic jog, where you have here the growth cone, and after some time it creates a rotation, so that it creates a turning so that the neuron can go to the proper direction, as if they could sense precisely where is the source. And this morphogen gradient can guide the development of axon over these large distances, probably due to this specific organization of the growth cone, where you have specific receptors that are interacting with the guidance cues and are transforming this positional information into a specific oriented motion. So in this review, basically we summarize this motivation. Then in the first part, we explain the modeling based on diffusion mechanism of the single cell sensing. So I will now briefly go over these different parts of the review. Then we will discuss the modeling of diffusion and computation of the fluxes at small hole. Then we will summarize how we develop this hybrid simulation and fast algorithm based on teleportation to avoid simulating in many Brownian particles. And then we will explain how we solve this triangulation sensing, which is the mathematical formulation of how you can reconstruct a source from the fluxes of small, the fluxes at small holes, small windows located on a piece of a cell. And then finally, there is in this review a discussion of what we think could be the biophysical principle of neuronal migration. So first part, diffusion mechanism of single cell sensing. So this is an example of a source here where the source generates uniformly a gradient of molecules. We have here a cell with 3 receptors, and based on the fluxes estimated on these 3 receptors, we assess the position or the velocity as a combination of the fluxes so that the particle can follow the gradient. And when the gradient is much more steep, the motion is going to be more directed toward the source. And so we will treat here the case of at least 2 and 3 receptor and more. So over the last 30, 40 years, the general trends in the field was to look at the question that was previously asked in this Brownian paper, 1977 in Biophysical Journal, where basically the author wanted to relate the local concentration near the cell to the arrival time of Q's in this ball of radius A when you have the concentration C, diffusion constant T, and they could relate this sampling time of molecule that can bind to this difference of concentration. And the problem has really motivated the field for all these years, looking at what happened when you have a fully absorbing sphere, partially reflecting, and ligand can be captured, recaptured, and so on. So this was really about understanding how the local concentration here could be related to the sampling time of these different particles. However, we have been interested in a question which is a bit different, and the question is the following. We have many Brownian particles that diffuse from a source, here x0. We have a reflecting cell except at 2 windows, 1 and 2. And what we are interested in is to calculate, to determine the probability of particle hitting one of these receptors 1 and 2, which can be summarized into this classical equation, Laplace equation for diffusion, Laplacian P equal minus Laplacian P equals delta Dirac, reflecting boundary condition on the sphere, except at these 2 absorbing holes. So from this, we wanted to estimate what is the probability to hit 1 before 2, which is related also to the flux of molecule at these 2 receptors. And then we ask the following question: if we know the fluxes here at 1 and 2, or if you have another one at 3, can we reconstruct the position of the source? And if there are fluctuations, at which resolution we can reconstruct x0? So this is discussed in the first part of the review. Then the modeling approach is to calculate the fluxes at receptors. So I briefly recall again what you can find in detail of the review, which is called the match asymptotics. This is how you calculate analytically the fluxes at each of these windows, and this is important because this will give us the possibility to invert the equation and to recover the position x0. So you have to solve minus Laplacian P equals delta, so there's a mistake here, it should be minus. So there are 2 possibilities. One is to find the solution far away from the windows, and so this allows to find the solution of the previous equation P using the Green's function. And here this is a boundary layer which comes from the inner solution, which is basically to tell us what is the solution very near the window, and that has to do in dimension 2 with the Green's function. So here you have Pi, which is an unknown coefficient to be found, and C is also an unknown coefficient. So this is a universal solution, and to find out this P and I, you have to explore what happened when the solution goes near the window xi. And also, you need to have your system of solution which has to be equal to 0 at exactly the boundary so of the absorbing window. So we have these match asymptotics. So again, for this, what happened far away and for what happened close to the window. So we have this general question of how to find this pi and this coefficient c. So for finding pi, you just go take the limit as the position x here tends to xi. You write the solution and it has to behave like the boundary layer, so this will give us a condition, and with this you obtain a system of n+1 equation for all the pi and 1 here for the total system of equations. So you have n+1, so pi is, you have n, and c is 1, so you can now solve your system of equation and find these probability coefficients. So this is an example that can be done in dimension 2, where you can calculate P2 and you can have, depending if your system is located on half a space or on a ball, you can express this probability in terms of the Green's function. So now, if you want to compare this analytical solution with numerical approach, what we wanted to do is to design hybrid simulation to avoid simulating all of these trajectories. So a typical case is that you have a source, you generate a Brownian trajectory, you look at if this trajectory will end in one of these two, and you can design your statistics of interest. If you do this, you have to wait quite a long time, especially if your domain is not bounded. We know in dimension 2 with probability 1 it will ultimately come here, but you have to wait basically an infinite number of time. So we decided to replace this with what you call a hybrid simulation, where instead of simulating the trajectory, we teleportate the initial source into a disk at a certain distance from the cell we were interested in. Then you can generate a Brownian simulation inside until it reaches a second circle, when then you would like to map it back to the first circle and then use Brownian simulation. So by this, you just generate Brownian simulation in a small region of interest. And this is an example how this is done here. This is a summary of this algorithm. For this, you need to calculate the flux to this prescribed abstract circle where you have to do your simulations. So then I would like now to discuss the triangulation sensing. So basically this is how we solved the previous equation with respect to the source x0. And we found out that it is possible to find, and for this you need to resolve this equation and have x0 as a function of x1, xn. And so if in dimension 2 you cannot recover the source, I mean in dimension 2 with 2 windows, if you have at least 3 windows, as you can see here, you need to find out this intersection of these different curves, and this intersection gives you the point where the source is located. And in addition, you can add a certain uncertainty of the fluxes, which you will find into the uncertainty region where the source is located. And as you can see, it depends on the source position. So the message, as we discussed in the review, depends on 3 receptors. And so you can see here again the example. With 2 you don't find anything. This is, you need the intersection of these trajectories. With 3 window you can. This is an example here. This is what happened when you have some uncertainty. And this is what happened when you have multiple sources, where you gain into the precision as you need to have this intersection of these different lines. And this is an example here where we have multiple of them. So then in dimension 3 you can recover the source location, and now you don't have any more the intersection of lines but of surfaces, and you need at least 3 surfaces to intersect into 1 point, and that's why we need 3 receptors as discussed here in this review. And so we design also an algorithm of how you can find the source when you have the fluxes through these 3 points. Basically, we go back to the equation that I've shown previously. For each receptors, we calculate the quantity of Fi squared, we minimize it, and the minimization corresponds to the intersection where the 3 of them are equal to 0. So then you can also calculate the difference of fluxes between the 2 receptors as a function of the distance to the source, and you can see how it decays. So this is well discussed in the review. So we define what is the sensitivity. Sensitivity is basically the difference of the fluxes of the probability between the 2 holes. But when you have 3, you have to take the maximum of the difference between them, and you define the region where sensitive is possible as the region where R is larger than as a given threshold. And you can see that it depends on the different localization, because if you have 2 points, they can either be together or they can be on the opposite diameter. So there are various possibilities that can be studied. So this is an example. If you put a threshold at 1, 5, or 10%, have the region where sensitivity, where you can basically detect your source, is possible. And so we also give estimates in dimension 2 and 3 that is can be related to the radius here of the cells. Epsilon is the size of the radius, and x0 is the distance from the source. So how much of this uncertainty? So we then ask the question, how is it possible to reconstruct the volume of uncertainty if you add, for example, a random noise into the fluxes? And this is examples of how this volume of uncertainty decays here when you increase the number of windows. So we have here this uncertainty radius, L is the distance to the source, and so we show here different quantities, such as how it increases with the distance L. Again, we can ask the same questions. You have this uncertainty radius. This is calculated here as basically what is the radius where 99% of the reconstructions of the correct reconstruction occurs. So when you have here the different sources, so we normalize here to the radius of the disk, which is 1, and you can see what is the effect of having uniform a single cluster or spread cluster into this reconstruction. And again, so we show here with the different cases with spread windows, single, and cluster, depending on the location of the source here, as you can see, how the cell is able to locate it where the source is positioned. And finally, the review discusses some principle of neuronal migration. So I thank you for your attention. And again, going from coordinate to the full wiring is still a very open question. I mentioned here some literature under which the review is based. Thanks for your attention.